Hello, everyone. My name is Leilani Tuani-Fifita, and today I'll be presenting the structure of skeletal muscle. In this session, we're going to move from the microstructure of muscle fibers to the broader organization of whole muscle, and then we'll connect those structural concepts to functional outcomes using a peer-reviewed research study. This structure to function connection is essential in physiology because how a muscle is built directly determines how it will perform. By the end of this presentation, you should be able to identify the hierarchical organization of skeletal muscle, describe the microscopic anatomy of the muscle fiber, explain the connective tissue layers that support and protect the muscle, relate structural features to functional capacity, and apply this knowledge using data from a real human study on muscle adaptation. Each of these pieces builds on the next, so by the time we reach the research section, the physiology we learned in the beginning will help explain the outcomes in the figures. So go ahead and get your pencils and papers and let's get into it. Our concept definition: skeletal muscle is a voluntary striated tissue, meaning we consciously control its contractions, and it displays a banded pattern under a microscope. This tissue is built from extremely long cells called muscle fibers. Those contain multiple nuclei because they originate from the fusion of many precursor cells. A key thing in skeletal muscle physiology is order. Every fiber contains repeating contractile units, sarcomeres. That allows the muscle to shorten efficiently and produce force. This highly structured organization is what allows skeletal muscle to continuously generate controlled, graded movement. When we look at the organization of skeletal muscle, it follows a very specific hierarchy. Whole muscles are divided into bundles called fascicles. Each fascicle contains many individual muscle fibers. Inside each muscle fiber are thousands of myofibrils, and each myofibril contains sarcomeres arranged end to end. Now, the sarcomeres hold the actin and the myosin filaments responsible for contraction. This means that the force produced starts at the sarcomere level. It's summed across the myofibrils, then across fibers, then fascicles, and then finally the whole muscle. This hierarchy provides both strength and precision, and those are two qualities that are essential for normal movement. The connective tissue structure. Now, in muscles, it's just as important as the fibers itself. The epimyosin surrounds the entire muscle and provides overall structural integrity. The perimysium wraps around each fascicle, organizing fibers into functional units. And the endomyosin encases individual fibers and contains the capillaries and nerves supplying them. So all these layers allow forces generated deep inside the fiber to be transmitted outwards to tendons and bones. Without these layers, the muscle contractions would not result in a coordinated limb movement. Now, fibromicroanatomy, excuse me. Now, when we zoom into the individual muscle fiber, we see several essential components. The sarcolemma is the outer membrane that conducts electrical signals. The nuclei, positioned around the edges, regulates protein synthesis for the large fiber. The mitochondria generated ATP needed for the contraction. They're the powerhouse. The T tubules carry action potentials deep into the cell. And lastly, the sarcoplasmic reticulum stores and releases calcium, which is what triggers the contraction. Together, these structures allow rapid coordination contraction throughout the entire fiber. Any dysfunction in these microcomponents can lead to the impaired muscle performance. Study introduction. So, to apply these structural concepts, we now shift into a peer reviewed research study that examines how resistance training influences muscle performance in youth populations. Now, understanding the structure is useful, but connecting it to real world outcomes such as strength and endurance or power is where physiology becomes clinically meaningful. Now, this study is going to give us an opportunity to see how changes at the sarcomere, fiber, and fascicle levels translate. We are analyzing the article titled Effects of Resistance Training on Performance in Youth Athletes by Granicher in 2016. This study is a systemic meta analysis compiling controlled trials involving children and adolescents aged 6 to 18. The authors examine how resistance training influences several categories of muscular performance. Those include strength, power, endurance, and sport specific ability. By comparing outcomes across age groups and modalities, the study helps us understand how the structural features of skeletal muscle translates into a measurable functional adaptation. In Grancher of 2016, Figure 2 summarizes the overall effects of resistance training. As I point here to the first bar, muscular strength, here at the top, it shows the largest improvement. This reflects increased myofibril density, greater sarcomere addition, and improved motor unit recruitment. Pointing here to the second bar, muscular power, it also increases significantly, which relates to enhanced calcium handling and faster cross-bridge cycling within the sarcomere. Moving on to our third bar, muscular endurance. This improves moderately because endurance relies on mitochondrial expansion and improved oxidative efficiency. And our last bar here, athletic performance. It's also a composite measure. This figure provides a foundation for figuring how structural adaptations manifest as measurable performance change. Still referencing Granicher of 2016, Figure 3 compares children and adolescents. Here we see on the left side, children show larger relative improvements. Children experience rapid neural adaptations, quicker recruitment efficiency, and a strong motor pattern development. You see here all the bars on the right? The adolescents also improve, but their adaptation are influenced by hormonal maturation, a larger baseline muscle size, and shifting fiber type proportions. Granicher and his colleagues emphasize that developmental stage shapes both the rate and the mechanism of structural response. In Figure 4 from Granicher of 2016, we see sex specific performance outcomes. As I point to these darker bars representing boys, they demonstrate larger gains in strength and power, reflecting higher initial mass, the slightly greater myonuclear accretion rates, which means a higher capacity for a protein synthesis during hypertrophy. Pointing to a lighter bar representing the girls, you see the improvements are still highly meaningful. Girls show strong neuromuscular adaptations and architectural strengthening, even with smaller absolute changes. And that also supports the author's conclusion that both sexes experience beneficial structural remodeling through training. In Figure 5 of Granicher in 2016, it compares training modalities. As I point here, you see that the complex training It yields the highest effects because it blends mechanical load and explosive movement, which together it stimulates a hypertrophy and neural refinement. Pointing to this middle set of bars here, we have the machine based training. It produces moderate gains by providing stable, isolated resistance. Now, lastly, we have our plyometric training, which enhances our power through improvements in tendon stiffness, elastic recoil, and rapid force development. Each training modality produces distinct structural adaptations, demonstrating that the type of stimulus shapes the type of muscular improvement. Now, integrating all results from Grancher of 2016, hypertr- hypertrophy emerges as a central mechanism of adaptation. Muscle fibers increase in cross sectional areas, sarcomeres strengthen their alignments, and connective tissue scaffolding becomes more robust. These changes enable greater force production and improve mechanical efficiency. The authors emphasize that age, sex, and modality influence the magnitude and nature of adaptation. Children benefit strongly from neural improvements. Adolescents exhibit more architectural remodeling, and all groups gain measurable structural integrity with appropriate resistance training. In summary, Granicher of 2016 reinforces that skeletal muscle is profoundly adaptable. Strength shows the most dramatic response. Power and endurance follow closely, and performance improves when training targets the structural characteristics of the muscle. This confirms the essential connection between muscle, structure, and function. These references here include Sherwood for our anatomical and our physiological foundations, and Graniger of 2016 for our scholarly data and informed our analysis of structural adaptation. Just wanted to thank everybody for staying with me through his presentation. If you have any comments, questions, or concerns, go ahead and let me know, and I'll be glad to get back to you. Everybody, have a great day.